Welcome back, everybody. I hope you had time to refresh yourselves. So this morning we had a very intense session where we discussed the design and the analysis of clinical trials. And I'm sure you all noticed that the emphasis was very much on evaluating the efficacy of drugs, determining whether they had the effect they were intended to have. We did not talk about safety, and this brings us to our second session. Obviously, safety of the drug products that come through our purview is a fundamental issue, a fundamental responsibility of FDA. Safety considerations change as we move through the drug development process. Early in the drug development process, we're concerned about the safety of trial participants. Later on, obviously, the concern becomes that of the general population exposed to the drug. The other thing that we didn't talk about this morning is that different groups in the population may respond differently to drugs. And that's another area which we're going to touch on in this session, which is special populations or subpopulations that need special consideration. So I'm delighted to welcome our speakers for this afternoon session. Shabnam Nasir, who will talk about the safety considerations in clinical drug development, and she'll be followed by Lin Yao, who will be talking about specific populations in clinical trials. So at this point, let me hand it over to Shabnam.
Hi, good afternoon, everyone. My name is Shabnam Nasir. As was stated, thank you for the kind introduction. I'm a lead physician in the Division of Anti-Infectives in the Office of Infectious Diseases. And my talk today will be focused on safety considerations in clinical drug development. I do appreciate having the opportunity to speak today. So our learning objectives include the following. We'd like to understand that protection of subjects should always be the first priority when designing early clinical studies. We'd like to learn key safety considerations in the conduct of phase 1 trials, and to understand the principles of safety monitoring and reporting in clinical trials. So the main objectives of phase 1 trials are to assess safety and tolerability of the drug product, attempt to characterize dose-limiting adverse reactions, determine maximum dose associated with an acceptable safety profile, to characterize pharmacokinetic parameters, and explore drug metabolism and drug interactions. The subjects in phase 1 trials generally tend to be healthy volunteers, and these volunteers have few confounding factors. In some instances, phase 1 trials are conducted in patients, and these are generally when the drug is known or expected to be toxic. An example would be a trial that's evaluating a cytotoxic agent. When patients are enrolled in phase 1 trials, there will be more confounding factors and sometimes more difficulty in trying to separate disease-related manifestations from adverse reactions. In addition, special populations such as patients with renal or hepatic impairment can also be enrolled in phase 1 trials. So here are some general considerations when one is moving from the non-clinical to first-in-human study. So we have to consider what is the evidence we've garnered so far from the non-clinical studies. And so we have to ask ourselves a number of questions. What is the duration and the total exposure proposed in humans? And how does that correlate with what was tested in the animals? We have to think about the characteristics of the test drug. Is it a small molecule? Is it a biologic? Does it have a long half-life? We have to consider the disease that's being targeted for treatment, because that would need to be taken into consideration in the benefit and risk assessment. We have to think about the population in which the drug will be used. Is the intent to enroll women of childbearing potential or children in the study? And what's the route of administration? Is the product being given systemically or topically? So these are some of the initial considerations. Do the non-clinical studies that have been done so far provide sufficient data to allow for the proposed clinical trial to be undertaken? So, what species were they studied in? Are the species appropriate and are they relevant to the particular drug? What are the potential target organs of toxicity? What was the duration studied in animals, and what was the dose and the route of exposure? Was it appropriate and similar to what is being proposed in the human study? And what about the PK and PD assessments? Were they appropriate? Was a dose response identified? And then again, do we need to do any special studies in animals if we're planning to enroll, for example, children or pregnant women? So it's important to keep in mind that only some toxicities that are seen in non-clinical studies translate into adverse reactions in humans, while there's other toxicities that do not. Excuse me. And so you end up with both predictable and unpredictable toxicities, and these can appear in any phase of development, and sometimes you actually only see them in the post-marketing setting. It really depends on the size of your safety database that was available pre-market. It's also important to keep in mind that certain adverse reactions are subjective, and these can be adverse reactions such as headache or other events such as hypersensitivity reactions that really can't be assessed in the non-clinical arena. So let's talk about some examples. So here's an example of predictable toxicity. And this example is from the anti-infective space where I'm from. I'm sure you can find other examples in other therapeutic areas. So Linezolid is an antibacterial drug that was approved in the late 1990s, and at that time it was the first member of the oxalidenone class. It had activity against gram-positive organisms and was also thought to have activity against some resistant organisms. So certainly there was some therapeutic benefit that could be offered by the product. The non-clinical studies that had been conducted showed that myelosuppression occurred, and this appeared to be both dose and time dependent. There were various changes in the bone marrow, and it appeared to affect all three cell lines. But the product had the potential to offer therapeutic benefit, and so further clinical development was pursued. When phase 3 trials were conducted, there was no evidence of myelosuppression per se, but there was a high frequency of thrombocytopenia that was seen in the linezolid treated patients. And at the time the product was approved, the prescribing information included precautions for thrombocytopenia, and they also noted a clinical, the animal pharmacology section described the hematopoietic effect noted in animals. After the product was approved, there were cases of myelosuppression seen affecting all three cell lines, and these were reported as leukopenia, anemia, etc. And so labeling was updated to include a warning regarding myelosuppression. Here's an example of unpredictable toxicity. So we had two products, both were members of the beta-lactam class, which is a very well-known class of antibacterial drugs, and we have a lot of experience in this class. The structure of these beta-lactams, however, had been modified to enhance the spectrum of activity. There were no unexpected toxicities seen in animals, so both of the sponsors proceeded to conduct their phase 1 studies. The single dose went well, but then when they went into the multiple dose trial, the subjects developed moderate to severe skin reactions, such that the product development actually had to be halted for both products. So in this case, this is an example of unpredictable toxicity, and even if the non-clinical studies appear benign and you don't see any adverse events, it doesn't mean that you're not going to see events when you conduct the study in humans. Now to a discussion on maximum recommended starting dose. And you will be hearing more about this in day 2 lectures, but I'll just go over some high-level concepts from the clinical perspective. So There are some key principles in selecting an MRSD. One is that you want to avoid toxicity at the initial clinical dose, but at the same time you want to allow reasonably rapid attainment of the trial objectives, and these are usually tolerability and PK, so that you're not so low that it'll take you forever to get where you need to be, where the target dose was. And typically we use an algorithmic approach based on what doses were given to animals and what toxicities were observed. There are some alternate approaches based on animal pharmacokinetics and modeling, but for the most part we use the algorithmic approach. Some of the key concepts when you look at the MRSD are the no observed adverse effect level, or the NOEL, and this is the highest dose tested in animal species that did not produce a significant increase in adverse effects compared to a control group. The second term is HED, or human equivalent dose, and this is really what is the dose in animals that converts to a certain milligram per kilogram dose in humans. Based on that, you would be able to come up with the starting dose. Then you also have to consider which animal species you use to make these calculations. Ideally, you would choose the most sensitive species, and that's the species from which you can identify the lowest HED. In some cases, however, you would want to use the most appropriate animal species because that species actually might have the particular target of interest, and this is especially seen with biologics. So here's a schematic that shows the steps in determining the maximum recommended starting dose, and for your reference, the guidance is at the bottom of the slide. So first you would determine the NOEL, which is a starting point for determining a safe starting dose for new molecular entities in healthy volunteers. Then that gets converted into the HED. You select the HED from the most sensitive or appropriate species, and then you choose a safety factor. Then you divide the HED by that safety factor to come up with a maximum recommended starting dose. So what is the safety factor? It's a number that gives you a margin of safety for protection of human subjects when you're moving from animal studies to humans receiving the initial dose. The default number is 10, but there is some degree of flexibility, and sometimes we can go higher or lower than 10. The safety factor provides us some reassurance that when we're moving from animals to humans, that we're not exposing human subjects to any great risk. One knows that there's variability when you move from animals to humans, and the safety factor allows for that variability to be accounted for. So, as I mentioned previously in the examples, there are uncertainties when you go from animals to humans. There can be differences in the sensitivity between animals and humans to certain products. There can be difficulty in detecting certain toxicities in animals, such as headache, myalgia. There could be differences in receptor densities or affinities. You always have to be prepared for unexpected toxicities. In addition, there could be intraspecies differences in how the product is handled, such as ADME. And keep in mind that, again, humans and animals may not always handle the product in an identical manner. So when would we have to increase the safety factor greater than 10? If it's a brand new class and we have no prior experience with the product, you might have to consider increasing the safety factor. Other issues could be that the toxicities in the non-clinical studies were severe or irreversible, or they could be toxicities that are non-monitorables. For instance, if you have histopathologic changes, but they were not monitored because there's no clinical methods to monitor those toxicities, or there are no biomarkers, that could be a reason to consider increasing safety factor. If there's a very steep dose response curve, that could indicate a greater risk in humans. If the PK is not linear, it limits the ability to predict dose-related toxicity. Also, if there's variable bioavailability, because you could have poor bioavailability in animals, and that may underestimate the toxicity when you move to humans. On the other hand, you could decrease the safety factor to less than 10, and some of the reasons would be if you're dealing with a member of a well-characterized class of drugs, if the toxicity is easily, that's produced is easily monitored, if it's reversible, if it's predictable, and also if the NOEL was determined based on toxicity studies of a longer duration, you might have a little bit of more reassurance in reducing the safety factor. So let's discuss an example of how you would calculate the MRSD. Here's a product for which the HED was about 400, derived from rats. The sponsor proposed a starting dose of 100 milligrams, so that brought the safety factor to 4. And the rationale provided was that this was a member of a well-characterized class of drugs, and that the toxicity studies in 2 species were of longer duration than what was proposed in the clinical trials. In addition, the potential toxicities were readily monitorable and reversible. However, we actually didn't agree totally with the rationale because we knew that members of the class had exhibited more toxicity than the parent class from which it was derived. And there was also some concern that the bioavailability in animals was low and that human bioavailability could potentially be greater than anticipated, leading to a risk of higher exposure in humans. So we actually agreed upon a starting dose of 50 milligrams, raising the safety factor to 8. So now let's move into some general considerations when you're going to be conducting these phase 1 trials. So more questions to ask yourself when you're getting ready to conduct these trials. One is, are the clinical trial protocols designed appropriately to ensure safety and meet the stated objectives? You have to protect the safety of the subjects in the trial as always. And do you have enough information about the quality of the investigational products? For example, has the formulation been well characterized? Do we know, for example, what the impurities are? Are the route and the rate of administration appropriate? Can the product be given as a bolus dose, or are there toxicities such that we would have to give it as a slow infusion? What is the mechanism or mode of action? Is it novel? Do we know anything about similar products and that act via a similar mechanism? Are you expecting an off-target effect? And that's a very important concept, especially if you're dealing with a biologic and the mode of action involves a target which is connected to multiple signaling pathways or affects biological cascade or cytokine release. Let's go into safety considerations regarding dosing. How do we determine an appropriate dose? Ideally, one subject should receive a single dose, and that would be followed by sequential administration within the cohort. You have to ensure that there is an adequate period of observation between dosing so that one has the time to observe and interpret adverse reactions in the cohorts at the lower dose. And how long you actually observe will depend on the properties of the drugs, the PK properties, how long it stays in the system, how long its half-life is. And so we also learn good lessons from previous trials conducted with similar products. So moving on to dose escalation. If the protocol for these phase 1 trials allows for dose escalation, you have to ask whether the dose escalation scheme is appropriate. Are the dose increments appropriate? Is it okay to double up or go up by 4 times? You have to be cautious and use a slower rate of dose escalation, especially if you have a small therapeutic window, if you had animal models that were not very good, or if you had any concerns about toxicity. There is a fair bit of flexibility here. Again, it just depends on your product, the target, and the toxicity found in animals. You have to ask yourself if the amount of information before each dose escalation is appropriate, and also if the number of subjects at each dose is appropriate. So next we'll move to safety monitoring. It's important that protocols have an appropriate monitoring scheme to look at clinical signs and symptoms of adverse events likely to be associated with the drug. And the duration of the clinical observation should also be adequate with respect to the stated objectives and endpoints, the anticipated response to the product, and the health-related conditions that are being studied. Sometimes we do need to monitor for a longer period of time, and again, it depends on the characteristics of the drug. Where we monitor also matters, whether it's a hospital setting that's needed. And if you're worried about any sort of delayed onset reactions, undetected toxicities, you have to make sure that the follow-up is long enough. So as far as frequency of monitoring, there may be a need to monitor more frequently soon after initial dosing in the first week or so. Certainly you would need to have follow-up visits for subjects who develop adverse events or laboratory abnormalities. In addition to monitoring clinically for adverse reactions, you'd also monitor laboratory tests. And then which particular laboratory tests you order and collect the data on really depends again on your drug and the target organs of toxicity. You want to ask yourself if the laboratory tests would include routine assessment of all organ systems. Do we know enough about the drug such that you only need to assess specific organ systems? Are they sufficiently detailed laboratory tests that you can actually make an assessment of whether or not the drug is causing toxicity of those specific organs? And then lastly, are there stopping rules for patients whose laboratory test abnormalities reach a certain threshold? So what are stopping rules? It's important that the protocol have clear stopping rules for administering the drug, enrollment, dose escalation, and rules for termination of the trial itself. Oftentimes, we've actually had to put INDs on hold because there were no stopping rules in the protocol. So again, these are changes to the protocol that are to be implemented when toxicity is observed. And to be able to have stopping rules, you have to identify what might be acceptable toxicities and what are not acceptable toxicities. Since it's unlikely that you'll have a drug with no toxicities, it's important to stop and try to generate these stopping rules. If acceptable toxicities occur, these will be the ones that don't result in changes in how the trial is continued in terms of subject enrollment and dosing. If you have other toxicities that are not acceptable, you have to have a procedure in place for what you would do with the study. So let's go on to our challenge question 1. Which of the following is used to calculate the MRSD? And hopefully everybody will get this. And this is the Noel. All right, so let's move on to sort of the second half of my talk, where we talk about where we continue to talk about the evaluation of safety, but sort of in larger, later clinical settings. So safety evaluation is a central component in all stages of drug development. It's an evolving process. It depends on the stage of the product under development. For products that are marketed, the safety information is reflected in the product labeling. It's also called the prescribing information, or PI. While up-to-date safety information for products that are under investigation would be contained in the investigator brochure, or IB. So again, these would be non-clinical findings from in vitro or animal studies, clinical pharmacokinetic safety data. And for unapproved products, the investigator's brochure is equivalent to the prescribing information. So what are the sources of safety information found in the investigator brochure or the prescribing information? Non-clinical data, again, can include in vitro or animal data, as well as information about the structure of the product and the manufacturing process. Clinical pharmacological data can include information about PK/PD, And then safety data can come from clinical safety information from healthy volunteers and patients. Perhaps the most pertinent information, of course, will be the data from the clinical trials for the same indication, as these data are generally controlled for the patient population characteristics and the underlying pathological process, and also from trials for different indications. You can also have information in the IB from post-marketing experience and, IB or prescribing information from post-marketing experience, the published medical literature, and last but not least, safety profiles of drugs in the same class. So now, why is safety monitoring required in all clinical trials? The goal of safety monitoring in clinical trials is to identify, evaluate, minimize, and appropriately manage risk. And this will lead to our discussion of adverse event ascertainment and monitoring. So we have a number of regulatory definitions that we'd like to go over, and I have given you the CFRs for your reference. Adverse event is any untoward medical occurrence associated with the use of a drug in humans, whether or not it is considered to be drug-related. This could be an unfavorable sign, symptom, disease, abnormal lab, a vital sign, or other testing, or a worsening of the above or a constellation of the above. Adverse events can also be referred to as adverse experience. Some examples of ways that adverse events can be ascertained include spontaneously reported or observed signs and symptoms. Symptoms are signs that are reported as a result of a probe, such as a questionnaire or a checklist. And then testing of vital signs, laboratory studies, and special safety assessments can also be conducted, and these can include, for example, visual, hearing tests, or EKGs. Upon reporting, adverse events are characterized according to their severity, and there are a number of severity grading scales or tables that are available to provide guidance to sponsors or investigators for safety monitoring in clinical trials. These are optional tools. They are specific to the study population that's being evaluated, for example, patients or healthy volunteers. Clinical abnormalities may be represented as either mild, moderate, severe, potentially life-threatening, for example, in healthy volunteers. If you have oncology patients, AEs could be graded by a scale of 1 through 5, where 1 is mild symptoms to grade 5, which is death related to an AE. So again, they're specific to the study population. And it's important to keep in mind also that here are some examples. There's probably more. So again, it's important to keep in mind that the term severe is not the same as serious. What happens when an AE report gets to the sponsor? So the process includes conversion from the investigator's verbatim terms to the standard preferred terms using a specialized dictionary called MEDRA. MEDRA is the Medical Dictionary for Regulatory Activities, and it can be used to sort AEs and group-like events. Terms are used to calculate the incidence of AE. Here's a schematic to illustrate the MEDRA hierarchy. It starts with the highest level of terminology, and that's the least specific, which is the systemic organ class, or SOC. And you move down all the way to the lowest level terms. This is in order of increasing specificity. The PT represents a single specific medical concept. So clinical investigators play a really crucial role in the safety reporting, and you would want them to use the most scientific and consistent terminology when reporting adverse events, as coding problems could result in splitting or lumping adverse events and therefore missing safety signals during the safety data analysis. So splitting similar terms such as hypertension, high blood pressure, these can result in a lower incidence, or they could minimize or mask a safety signal. And on the other hand, lumping dissimilar terms to the same PT could obscure a safety signal under a lumped term. So for example, facial edema is very different from leg edema as far as pathophysiology, and a coding issue could be lumping all edema to the PT edema. In this third bullet, there could be a lack of an adequate term or definition to represent a syndrome of interest such as drug hypersensitivity or serotonin syndrome. And in this case, you may need a prospective case definition if the syndrome is not well characterized by a single term. Moving on to another regulatory definition of serious, again, not to be confused with severe. Serious adverse events are any adverse events that, in the opinion of the investigator or sponsor, results in the following. So death or life-threatening conditions with the immediate risk of death, in hospitalization or prolongation of existing hospitalization, a disability or a birth defect, and last but not least, important medical events. And these would be those that could jeopardize the patient or require intervention to prevent one of the outcomes listed above. Examples may be allergic bronchospasm, which would require intensive treatment in an ER or at home. So here's a list of examples of uncommon SAEs. And if you see one of these, it could raise a red flag that there might be a possibility that the AE was caused by the study drug, especially if you have a study population of generally healthy individuals. You really shouldn't see one of these SAEs. This is not a comprehensive list, but it may be a good start. So on this slide, I'll discuss points to consider for investigators in evaluation of SAEs. SAEs are, like any other adverse event, need to be assessed for drug-related causality. And you can do that by asking a number of questions. One would be, is it common? Is it a common occurrence in the population under study? Was it treatment emergent, meaning did it occur on treatment or shortly thereafter? Did it respond to drug discontinuation or de-challenge? Did it reoccur upon introduction of the study drug or re-challenge? Were there concomitant medications? Were there any pertinent laboratory studies or other tests done to try to characterize the adverse event further? Was there an obvious alternative cause? And lastly, but not least, was the SAE a study endpoint? For example, if death occurred, was that also a study endpoint? So you also want to report adverse events by providing enough relevant clinical information in the case report form. You want to include enough information in the case report form to allow a good quality narrative. So now let's talk about what the clinical investigator reports to the sponsor. And again, the CFR is there for your reference. So an investigator must immediately report all SAEs whether or not the event is considered drug-related. The investigator must also include an assessment of whether there's a reasonable possibility that the drug caused the event, and this is usually called a causality assessment. Study endpoints that are SAEs must be reported in accordance with the protocol. One exception is that if there's evidence suggesting a causal relationship between the drug and the event, then in that case the investigator must immediately report the SAE to the sponsor. In addition, the investigators should also record non-serious SAEs and report them to the sponsor according to the timetable that's noted in the protocol. So let's discuss a hypothetical case. I think that this might illustrate some of the points mentioned on the last couple of slides. This is case 1. You're an investigator for a clinical trial evaluating whether antihypertensive drug A is associated with a reduced risk of death, MI, or stroke, and death is a study endpoint in the trial. You're told that an 80-year-old white male just died in the trial, and we're also actually told the cause of death was anaphylaxis. So the question is, do you have to immediately report this case to the sponsor? So let's take a minute to think about what to do here. So you're told that death is a study endpoint, and usually an SAE that is also a study endpoint can just be reported per protocol. However, here we're told that the death was due to anaphylaxis. So if you recall in the previous slides on uncommon SAEs, anaphylaxis was listed, and with that SAE, that should be sort of a red flag that there could be evidence to suggest a causal relationship between drug A and event, and so the investigator must immediately report this event to the sponsor. So the question is yes. The answer is yes to this question. All right, so moving back to some regulatory definitions, we'll discuss the ones important to expedited reporting of adverse events to FDA. And the concept of unexpected adverse event is presented here. An adverse event is deemed unexpected if it's not listed in the investigator brochure or IB, or if the IB is not available or required, if it's not consistent with the risk information described in the General Investigational Plan or elsewhere in the current application. It would be unexpected if it's listed in the IB, but it's not at the specificity or the severity observed. For example, the IB lists increased creatinine, but you have a case of acute renal failure, and that would be considered unexpected by virtue of greater severity. Other examples would be if you have a cerebral thromboembolism, and this would be considered unexpected by virtue of greater specificity if the IB listed only CVA or cerebrovascular accident. And it could also be considered unexpected if it's mentioned in the IB as anticipated due to the pharmacological properties of the drug, or if it occurs with other drugs in the class, but not with the particular drug under investigation. So one example would be angioedema. It's anticipated to occur in patients that are exposed to the class of ACE inhibitor drugs. Angioedema would be described in the IB as a class effect, but a case of angioedema observed with the drug under investigation would still be considered unexpected for reporting purposes until it's included in the IB as occurring with the drug under investigation. Go on to another hypothetical case, case 2. Here you're the clinical investigator for a trial evaluating a new anti-malarial drug B for the treatment of acute uncomplicated malaria. The investigator brochure lists elevated hepatic enzymes. And the question would an event of hepatic necrosis in this trial for drug B be considered an unexpected AE? So I'll give you a moment to think. So the answer is yes, hepatic necrosis in this hypothetical case would be considered unexpected. And the reason is because the IB does not list the AE at the severity observed. So you're seeing hepatic necrosis when only elevated hepatic enzymes are noted in the investigator's brochure. Okay, so moving on to our next definition, suspected adverse reactions. So a suspected adverse reaction is an adverse event for which there's a reasonable possibility that the drug caused the adverse event. Again, reasonable possibility means that there's evidence to suggest a causal relationship between the drug and the AE for purposes of IND safety reporting. So let's look at some examples of suspected adverse reaction. This could be a single occurrence of an uncommon event that is known to be strongly associated with drug exposure. So for example, angioedema, Stevens-Johnson syndrome. Another example would be one or more occurrence of an event that's not commonly associated with drug exposure, but is otherwise uncommon in the exposed population. So if you have healthy subjects and they're developing neutropenia, that could be an example of this, or a heart valve lesion in a young adult, or tendon rupture is another example. And another type of example is an aggregate analysis of specific events observed in a clinical trial, indicating that those events occur more frequently in the drug treatment group than in a concurrent or historical control group. And an example of this would maybe having several acute myocardial infarctions in long duration trials of elderly cancer patients. So overall, the determination of an AE as a suspected adverse reaction it can be difficult, but it's really critical to avoid submission of uninformative safety reports. And the sponsor is the one that should evaluate all available information and decide whether there's a reasonable possibility that the drug caused the AE. And that's because the sponsor is actually the ultimate party that determines if a drug is or is not causally related to the AE. Here I wanted to show a slide that illustrates the universe of suspected adverse reactions. So these are actually not just adverse events with undetermined causality, and adverse events here are noted in the outermost yellow sphere, but they're not yet definitely established adverse reactions, which are noted in the dark red sphere in the center. So suspected adverse reactions are actually in the middle. Adverse reactions are a subset of all suspected adverse reactions where there's a reason to conclude that the drug caused the event. Now let's talk about IND safety reporting by the sponsor to the FDA. FDA continues to require that 3 criteria generally be met before a sponsor submits an IND safety report. And again, the CFR is provided here for your reference. So namely, the event must be serious, unexpected, and there must be evidence to suggest a causal relationship between the drug and the event. Often this standard is called a SUSAR, and it stands for Serious and Unexpected Suspected Adverse Reaction. So again, the sponsor must assess whether or not there's a reasonable possibility that the drug caused the event, and the sponsor should only submit these type of IND safety reports when all 3 criteria are met. In addition, sponsors are expected to submit an annual report in the IND that includes a summary of the most frequent adverse events. It should also contain a summary of the SAEs. So now let's talk about timeframes when it comes to IND safety reports. These reports are required to be submitted to FDA in 15 days, and those that are required to be submitted within 7 days. As you can imagine, the most serious events must be reported within 7 days. And there are 5 categories of adverse events that trigger the sponsor's responsibility to submit an IND safety report. One is the SUSAR that we just mentioned, serious and unexpected suspected adverse reactions. Findings from other studies suggesting a significant risk in humans. Findings from animal and in vitro testing suggesting a significant risk in humans. An increased rate of occurrence of serious suspected adverse reactions, and serious adverse events from bioavailability and bioequivalence studies not under IND. And sponsors have to submit IND safety reports within 15 days of the adverse reaction unless the adverse reaction was fatal or life-threatening, in which case the report must be submitted to FDA within 7 days. And there may be additional details that you might want to look at in the guidance that I've provided in my references regarding the timeframes. So in summary, I've tried to provide a high-level overview of safety considerations in phase 1 trials, important questions to ask yourself before you decide to dose humans as you translate the information from non-clinical studies to either healthy volunteers or patients, what's the relevance of toxicities seen in the non-clinical studies, and how they translate to adverse events in humans. It's important to keep in mind that there are both predictable and unpredictable adverse reactions. We also discussed how to go about determining a safe starting dose in humans and the concept of the safety factor. We next discussed the importance of safety monitoring and stopping rules, and the ascertainment of safety in larger clinical trials, and some important regulatory definitions of adverse events and monitoring. Finally, we also discussed how investigators play a really integral role in assuring quality safety assessments and the requirements of reporting to the sponsor. And then in turn, we discussed about how the sponsor will evaluate all available safety information and report to the FDA and all participating investigators. Okay, so this brings us to our last challenge question. This is a true or false question. If the adverse event meets all three of the definitions for suspected adverse reaction, serious, and unexpected, it should be submitted as an IND safety report. So I'm going to give you a few seconds to think about it. So the answer here is true. I hope everyone got this one. All three criteria should be met for it to be submitted as an IND safety report. Now I just wanted to show you my references. If you had any more questions, please refer to these. And I thank you so much for your time today. Appreciate the opportunity to speak. And the next speaker will be Dr. Lin Yao, who will present on special populations. Thank you very much.